Video opens with slow-motion footage of a prism splitting a beam of light into a vibrant rainbow. Against a dark, dusty background, the sound of subtle glass clinking and a low, resonant hum. How far would you go to understand a rainbow? For some of the greatest minds in history, the answer was uncomfortably far. To chase the fleeting nature of color, early scientists didn't just look at the world. They turned their instruments inward, upon the most delicate of organs, their own eyes. They willingly stared into the sun, painted their corneas with toxic chemicals, and even, in one famous case, risked permanent blindness for a momentary glimpse into the mechanics of perception. These weren't just experiments, they were acts of radical, sometimes reckless, self-sacrifice. And in their quest to decode the spectrum, they laid the foundations for everything we know about how we see. This is the story of the forgotten, and often dangerous, experiments that forever changed our vision of color. So, what is color really? And what happens when someone who has never seen it suddenly can? The dream of restoring sight is as old as blindness itself, but the specific quest to restore color vision to the colorblind is a surprisingly modern chapter. Before we could even conceive of such a feat, we first had to understand what it was we were trying to fix. The earliest documented attempts at sight restoration weren't about color, but about light and shadow. In the 18th century, surgeons like Jacques Daviel in France pioneered cataract extraction, a terrifying procedure in a world without anesthesia. Patients, held down, would have a needle inserted into their eye to break up the clouded lens. When it worked, it was a miracle. A world of monochrome shapes would flood into a mind that had known only darkness. These operations, while not restoring color in the way we think of today, were the first critical step. They proved that the eye was a mechanical system that could, in theory, be repaired. They gave future scientists the audacious idea that if you could fix the lens, perhaps you could one day fix the very cells that perceive red, green, and blue. But the path to understanding color was paved with more than just medical ambition. It was colored by some of the most bizarre and dangerous materials imaginable. The palettes of the past were a minefield of toxicity. To study color, scientists and artists alike handled pigments that could kill. There was mummy brown, a rich brown paint made from the actual ground-up remains of ancient Egyptian mummies, a ghoulish pigment used for centuries. Then there was emerald green, also known as Paris green, a vibrant hue beloved by Impressionist painters. Its secret ingredient was arsenic. It flaked from wallpaper, poisoning entire households, and was a favorite tool for assassins. Scientists of the era, in their poorly ventilated laboratories, would grind these pigments, mix them, and observe their properties, unknowingly exposing themselves to heavy metals and carcinogens. They were literally dying for their art and for their science, surrounded by the very colors that were slowly, silently destroying them. This willingness to risk one's own body for knowledge reached a shocking peak with none other than Sir Isaac Newton. While his work with prisms is legendary, the moment he fractured white light into its constituent colors, a lesser-known experiment, reveals the true depth of his obsession. He wanted to understand not just the nature of light, but how the eye itself physically responded to it. So, he took a bodkin along, blunt needle used for sewing, and slid it between his eyeball and the bone of his eye socket. He then pressed on the back of his eye with the tip of the needle. Why? He wanted to see what would happen. In his own words, he reported seeing several white, dark and colored circles which would appear and change as he manipulated the pressure. He was inducing phosphenes, the phenomenon of seeing light without light actually entering the eye. It was an act of breathtaking recklessness, a self-experiment that could have ended in hemorrhage or blindness, all to prove that our perception of color was a physical, mechanical process happening inside our own heads. Newton wasn't alone in his hands-on approach. A century later, Thomas Young, a polymath who helped decipher the Rosetta Stone, turned his attention to light. He proposed the revolutionary idea that the eye must contain just 3 types of color receptors, sensitive to red, green, and violet light. To test his theories, he performed his own, less invasive, but equally obsessive experiments. He would spend hours in darkened rooms, staring at filtered light sources, meticulously documenting the afterimages that burned into his retinas. By fatiguing one set of his color receptors and then looking at a white surface, he could see the complementary color, providing evidence for his trichromatic theory. Following in his footsteps, Hermann von Helmholtz refined this work, developing what is now known as the young Helmholtz theory. He, too, used his own eyes as a laboratory, building elaborate contraptions with prisms and colored glass to precisely measure the limits of his own color perception, pushing his vision to its breaking point to map its boundaries. The Victorian era saw this spirit of self-experimentation evolve into something even more direct, and arguably more disturbing. As medical science progressed, some doctors became convinced they could treat eye diseases by applying chemicals directly to the eyeball itself. Convinced that certain conditions were caused by a stain or imbalance in the eye as humors, they would paint patients' eyes with substances like silver nitrate. While sometimes used as an antiseptic, in other cases, it was a desperate, theoretical attempt to chemically alter the eye's function. The goal wasn't always clear, but the method was brutal. These experiments were often based on flawed theories, but they reflected a growing belief that the eye was a chemical, as well as a physical system. The pain and potential damage were immense, yet they were another strange, Dark stepped towards the idea that the biology of vision could be directly manipulated. It was amidst this flurry of research that one of the most fundamental aspects of color vision was finally recognized: the fact that not everyone sees it the same way. The chemist John Dalton, a contemporary of Thomas Young, noticed something odd. The color of a geranium flower, which appeared vibrant pink to others by daylight, looked sky blue to him. By candlelight, it appeared red. He meticulously documented his and his brother's unusual perception, realizing they couldn't distinguish between red and green. In 1794, he presented the first ever scientific paper on what we now call color blindness, which he hypothesized was caused by a discoloration in the vitreous humor of his eye. He even willed his eyes to science for dissection after his death to prove his theory. He was wrong about the causes in the retinal cells, not the humor, but he was the first to properly identify and study the condition. This discovery led to the development of the first color blindness test, like the Ishihara plates, those familiar circles of colored dots with a hidden number inside, designed to distinguish those with normal vision from those, like Dalton, living in a different chromatic world. Today, the spirit of those early, daring experiments lives on. But with technology that Newton and Dalton could scarcely have dreamed of, we no longer need to stick needles in our eyes. Instead, we can use the most sophisticated tool of all, genetics. Gene therapy is achieving what was once science fiction, restoring color vision to the colorblind. The condition of achromatopsia, a rare genetic disorder where individuals see the world in complete grayscale, is caused by faulty genes that prevent cone cells in the retina from functioning. In recent groundbreaking trials, scientists have used a harmless, modified virus to deliver a correct copy of the faulty gene directly into the retinal cells of patients. The virus acts as a microscopic delivery truck, inserting the new genetic code and allowing the cone cells to, for the first time, produce the proteins needed to detect color. It is the modern echo of painting the eye with chemicals, but instead of a blunt instrument, it is a procedure of unimaginable precision, and the results are nothing short of miraculous. Imagine living your entire life in a black and white film, and then, one day, someone flips a switch. In 2023, a young boy with achromatopsia who received the therapy described his first experience of color not as a single event, but as a cascade. The red of a toy car wasn't just a new shade of gray. It was an entirely new sensation, a feeling. Patients in these trials describe the overwhelming, sometimes confusing, joy of seeing a green lawn, the subtle hues of a sunset, or the specific shade of a loved one's eyes for the very first time. One patient, a man in his 50s, wept upon seeing the vibrant colors of a fruit stand. He had known what a banana was for 50 years, but he had never truly seen yellow. These stories are a powerful testament to the profound connection between color and emotion. They are the culmination of centuries of curiosity, of reckless experiments and brilliant theories, all leading to this moment where a life of monochrome can finally blossom into a world of full, vibrant color. From Newton's bodkin to a virus carrying a corrected gene, the journey to understand color has been a relentless human endeavor. It has taken us from dusty labs filled with poison pigments to sterile operating theaters, performing genetic miracles. It's a story of wonder and madness, of brilliant minds who risked their own sight so that we might better understand ours. And it leaves us with a final, lingering thought. If we can now give the gift of color to those who have never had it, what other senses, what other perceptions are waiting to be unlocked? What other colors, beyond our own limited spectrum, are waiting just beyond the edge of our vision, ready to be seen? Perhaps the next great experiment is already underway. And as always, thanks for watching. Video closes with a slow zoom out from a modern gene therapy lab. The image of a DNA helix slowly morphing into a twisting, colorful rainbow. The screen fades to black.